Now we have increasing resistance to antimicrobial medication. Unfortunately, because we're using more of these medications and we're not using them correctly, this is encouraging microorganisms to evolve resistance. So when penicillin G was first used, only 3% of staph organisms were actually resistant. Now more than 90% of Staph aureus cultures are resistant. This means that we're using hundreds of tons of penicillin to no good effect each year. The rise in resistance is a problem because it makes medications more expensive, there's complications, it's harder to treat. And dealing with the problem requires understanding the mechanisms and spread of resistance. So remember, we do have spontaneous mutations that can occur during replication. These happen at a very low rate, but they can have a significant effect. Just changing one base pair can actually cause a protein to become resistant to streptomycin, where streptomycin would normally target the protein. When you're looking at a population of 1 billion cells, at least one of them is going to have that mutation. So if you add streptomycin, you're going to kill everything but that cell. The problem is that cell is going to undergo binary fission and create a colony, and all of those cells are going to be resistant. Now, spontaneous resistance to antibiotics with several targets is less likely. This is why a lot of times we now use combination therapy with more serious diseases, because if you use multiple antibiotics, the chances that the cell will have simultaneous mutations to allow resistance to multiple drugs is very unlikely. You can also have gene transfer. So remember, you can have conjugation occurring where cells are sharing R plasmids with each other through those sex pili, and usually R plasmids do have several resistance genes. You can also have spontaneous mutations. When you look at soil microbes that are actually producing antibiotics, they can actually have these enzymes that are resistant so it doesn't poison themselves, but they can then spread them to other bacteria as well. Unfortunately, we have a lot of resistance that is emerging and becoming worse and worse. So if you look at the enterococci, they're part of the normal intestinal microbiota. They are a common cause of healthcare-associated infections. They are intrinsically less susceptible because they're gram-negatives, right? However, vancomycin is an antibiotic of last resort, and a lot of them have R plasmids that are allowing them to be resistant to vancomycin, and they can share those R plasmids with other bacteria. The Enterobacteriaceae are resistant to a lot of antibiotics. Remember, gram-negatives have that outer membrane to prevent entry of a lot of medications. They can also produce different types of enzymes now that actually will target antibiotics to break them down. When you look at Mycobacterium tuberculosis, it requires a long treatment period. A lot of times you can have resistance to first-line antibiotics through mutations, and that's why they now use combination therapies to try to prevent multiple mutations from forming. Typically, you need at least 6 months of treatment to get rid of it because they do grow so slowly, so it takes a long time for the drugs to actually have an effect. When you have multi-drug resistant TB, it's resistant to 2 of the first-line antibiotics, isoniazid and rifampin. And with extensively drug-resistant TB, it's even more of a problem because it's actually becoming resistant to second-line antibiotics. And remember, those second-line antibiotics are the antibiotics of last resort. Gonorrhea used to be susceptible to penicillin. Now it's not. Some have mutations, others have acquired plasmids. Only certain cephalosporin medications are actually effective right now, so you have to use combination therapies with gonorrhea. Also, Staph aureus is becoming resistant. We've all heard of MRSA, methicillin-resistant Staph aureus, but new strains are emerging. When you look at healthcare-associated MRSA, it is actually resistant to more medications than community-acquired MRSA, which is more easily treated. And from healthcare-associated MRSA, we've had 2 new strains form, vancomycin intermediate Staph aureus, VISA, and vancomycin-resistant Staph aureus, VIRSA. Remember, vancomycin is the antibiotic of last resort. If that stops working, we're in trouble. We also have Streptococcus pneumoniae, it is beginning to gain resistance to drugs like penicillin. So in order to slow the emergence and spread of resistance in bacteria, we actually need worldwide cooperation. It's the responsibility of physicians and healthcare workers to not just prescribe antibiotics willy-nilly. You have to educate patients about how to take them properly. Only give antibiotics if they're appropriate, right? Antibiotics don't work against viruses. Also, try to actually identify the infection so you can give something that's narrow spectrum, not broad spectrum. In terms of being a patient, you have to be a responsible patient. Follow the instructions even if they're inconvenient. You have to take the antibiotic as frequently as they tell you to. Otherwise, the levels get too low in your blood and it's not actually killing all the organisms. Ones that are less sensitive can actually grow and spread at that point. You know, antibiotics, again, they're not effective against viruses. They can't do anything against cold and the flu. And it's misuse that is causing this rise of antibiotic resistance. Unfortunately, we're having worldwide effects from this. Bacteria don't recognize, well, this is the United States, and that's Mexico, and that's Germany, and that's Egypt, right? They just spread. And antibiotics in other countries are available without a prescription, which has led to them being used incorrectly. Also, if you look at a lot of our animals that we eat for food, they have antibiotics in their food at low levels, which selects for antibiotic-resistant microbes. There is a lot of antibiotic-resistant salmonella now appearing because of this. Now, when we're looking at medications to target viruses, viruses are harder to target. Remember, viruses basically hijack the cell to do everything they need. They're just nucleic acids surrounded by protein. Now, they can code for different forms of polymerases, which are a potential target, and you can try to target things like replication within the cell. But when you look at all of our current antivirals, they are specific against a specific viral process in a specific virus, and we do not have any medications that can deal with latent viral infections. So what can you do? Well, you can try to prevent viral entry. If you can't enter the cell, you can't do anything. You can interfere with viral encoding. So not only do you need to enter, but you have to separate the nucleic acid from the protein coat. You can interfere with nucleic acid synthesis. Well, if you're not replicating the DNA or the RNA, the virus can't do its thing. So you can actually interfere with this. The problem is that you can have significant side effects because guess what? We also have nucleic acid synthesis happening normally in our cells. You can try to prevent genome integration. So this is used with HIV. You try to prevent that provirus from occurring. So if you can prevent HIV from its DNA integrating with our chromosome, you can try to prevent infection. You can also try to prevent assembly and release of viral particles. So there are enzymes required for maturation to take place. So you have medications that can actually prevent that process. Now, when you're talking about eukaryotic pathogens, one of these groups will be the fungi. It's important to realize that it's very difficult to target eukaryotic pathogens because they resemble human cells. They're more closely related than we are to bacteria. So a lot of antifungals will actually target plasma membrane synthesis and function. So antifungals typically will target ergosterol, which humans do not have. But it depends on the medication you're using. Some of them are more toxic, and if they cause the cells to leak, that can actually have side effects to us as a patient. You can target cell wall synthesis because cell walls have chitin, which we don't have. You can target cell division. Now, what's interesting is even though we also have tubulin, for some reason the medication seems to be taken up at a higher rate by fungal cells than human cells. Therefore, it is actually more selectively toxic to the fungus. You can also target nucleic acid synthesis, but as we've described, nucleic acid synthesis targeting, it's not good. It's a poor target in bacteria, it's a poor target in viruses, it's a poor target in fungi. You can also target protein synthesis. So you try to go after an enzyme that adds amino acids to tRNA. This is extremely toxic internally, but you can actually take it topically. So this is how we treat things like nail infections. Now, in terms of anti-protozoan and anti-helminthic medications, there's not a lot of research and development here. They're in poorer parts of the world, and to be honest, we just don't care. So you try to target biosynthetic pathways of the protozoans and neuromuscular function in worms.